Okay, thanks very much. I've just had a thumbs up from those who must be obeyed to tell me it's time to start. So my name's Ian McFadgen. I'm the Dean of Undergraduate Students here at the School of Biomedical Sciences. And it's my pleasure to welcome you today to this open day here on the Guy's campus. I'd like to thank you for taking your time out to visit us here. I think it's important when you're deciding about which university to go to that you actually take time to visit the place and get a feel for how the institution operates and whether you think you'd be happy here for 3-4 years. So please, please, please take this opportunity today to suck us dry of information and don't leave here feeling, if only I'd asked that question. I'd also like to thank you for applying to King's in the first place. We know there's lots of choice out there, lots of options open to you, so thank you for putting us down as one of your possibilities. And congratulations on getting an offer from us. So we're pleased that you're here, and hopefully we can give you a bit more information on what we have to offer. So why would you think about coming to King's College London? Obviously, you've been kind of through this to some extent already in making one of your 5 UCAS choices. This is a bit of kind of a marketing, but I guess we would say that we are what's sometimes called a Premier League university. We remember what's called the Russell Group of Universities, which are the 25 research-intensive universities in the UK. So in addition to teaching students, we also carry out research in those areas that we deliver education in, and that includes the biomedical sciences. And a big chunk of my time and my colleagues' time is spent researching into different aspects of the biomedical sciences. So sometimes we talk about having a research-informed curriculum where the curriculum that you follow is informed to a greater or lesser extent by the research that we are doing. And that particularly happens in later years in your programme. When you get to final year of your BSc programme, the modules that you will take are very much directed by the research that the staff are undertaking at the time. There'll be some areas that we don't do a lot of research in where we may not be able to deliver final year modules in, but those areas that we do research in, for example, cancer research, neuroscience, immunology, you will get lots of options in those kind of modules. So the kind of flavour of the degree that you get, whether it's at King's or any other university, is governed to some extent in later years by the research that's going on within the walls of the university. In addition to teaching biomedical sciences, we teach a whole host of other health-related degree programs: medics, dentists, physiotherapists, nurses, midwives, pharmacists, dietitians. So we're actually one of the largest health education centres in Europe, probably the largest in Europe. We've got close relations with our trust hospitals, so St Thomas's Hospital, Guy's Hospital, King's College London Hospital, and that means that the education you get is very much focused along a kind of molecules to man. As far as the biomedical sciences are concerned, and by that I mean the programs you've applied for, as far as the biomedical sciences are concerned, we really look at the transition of how an idea developed in the laboratory ultimately feeds through to advances in patient care, patient treatment. So in my particular discipline, how an idea for a new drug target might be developed in the laboratory and how that's fed through to development of a new drug to treat disease. So that molecules to man transition really runs through everything that we do, because that's what having this concentration of health and health-related programs allows us to do. We carry out fantastic research. About 60% of our research is world-class, and we're currently going through another research assessment exercise where the government is assessing what we do, and we're hoping this number will go up. But at the same time, the reason you're here is because you want to be taught in the biosciences. And I'm proud to say that our teaching is also related excellent, both by the Quality Assurance Agency, which is the government body, and also by our students. Our students each year fill in what's called the National Student Survey, and above 90% of our students are satisfied or highly satisfied with what we produce. And one of the reasons we think that we can maintain this excellence in research and excellence in teaching is that we have a policy that everyone here, every member of staff here, no matter how high-flying the research will be, they will be teaching students. So every researcher here is put in front of students and is asked to teach them. And that's an incredibly potent way to learn about your discipline. If you're doing a final year module which is being taught by the people whose papers you've been reading, who are really pushing forward the boundaries in that discipline, it's a great way to learn about it. You can ask them questions. You can ask them, well, where does this subject, what's the direction this subject is taking? Where do you think it will be in 5 years' time? It also makes you proud when you see your tutors popping up on TV now and again with opinions on whatever. So all of our researchers, even our best researchers, are teaching. And altogether, we produce these programs, and I'm glad to say that in addition to our students being satisfied with what we produce, the employers are also satisfied with what we produce. We've got a very high employment rate for our undergraduates, but 95% plus or thereabouts. And one of the reasons for that is because, as I'll mention later, we teach you about the subject matter surrounding your science degree, but we also teach you skills that you'll need regardless of what career pathway you choose to follow. So whilst many students will continue to follow science-based careers, may end up working in laboratories, in the pharmaceutical industry, wherever, others will follow careers which are really not much to do with science, but the actual skills that they learn, the ability to identify a problem, develop a hypothesis, think about how they might approach that problem, what kind of experiments they might have to carry out to address that problem. Those kind of ways of thinking, be able to produce reports, be able to communicate your ideas, those ways of thinking apply to whatever career you go into. And one of the things that's my personal mission is to try to get more scientists into kind of decision-making bodies in society. For too long, we've had scientists who feel they've got to stay in a lab environment wearing a white coat. But actually, we need to get scientists into those kind of decision-making policies. It'd be great for me, as a scientist, to find someone on TV talking about genetically modified crops who wasn't talking about second, third-hand information, but was actually a scientist who had looked at the scientific evidence, analysed the scientific evidence, and could give us a real kind of evidence-based opinion on the matter. So there is real room for this generation to get involved and can be decision makers of tomorrow with their science degrees. Another reason you might want to come to King's is because, as you probably know, as you're here, we're right slap bang in one of the world's great capital cities. We're right slap bang in the middle of London. That's great from the perspective of social life that our students enjoy. It's also great because, as I've already mentioned, we as academics in the college get involved in a various kind of decision-making bodies. So it's kind of easy for politicians, government organisations to not just nip down the road across the river from House of Parliament to get our opinion on various matters. So we are involved in those kind of decision-making bodies. And also within London, although you're going to be studying at King's College London, there are lots of other educational institutes within London, and I mean by that other universities, museums, various other opportunities where you can go along and learn about your discipline. So the Science Museum, for example, runs lots of courses for the general public, which students are able to go along to and learn about different aspects of the sciences. So it's a very potent environment to learn about the sciences and beyond, aspects outside the sciences, for example. So being in London is a fantastic place to be. Now, for those of you who are not from London, just to orientate yourself, so this is the House of Parliament, this is the River Thames, and currently we're sitting down here at the Guy's campus, which is actually right by the Thames because the Thames does a big bend. But there are 4 other campuses in this map. First of all, the Strand campus, which is just at the north end of Waterloo Bridge. That's a kind of historic centre of King's College London. We've got a campus at the south end of Waterloo Bridge called, strangely enough, the Waterloo campus, which is right by the IMAX cinema. We've got some teaching that goes on at St Thomas's Hospital, primarily for medical students, down at St Thomas's Hospital, directly across the River Thames. Now, for those of you studying the biomedical sciences, the 2 main campuses you'll be concerned with are the Waterloo campus and the Guy's campus. About 70–80% of your teaching will be on this campus, maybe 20%, depending on which modules you follow, will be here. If you decide to take a modern language, for example, we encourage all our students to take a modern language alongside their science modules. They are taught at the Strand campus. If you walk from Waterloo to Guy's by the direct route, it takes about 20 minutes, about 7 minutes on a Boris bike or a Barclay bike, whatever you want to call it. But if you walk around the river, past the Tate Modern, past various distractions, it can take you several days to arrive at your destination. Now, those of you who are looking at this will realise that this is out of date because in the past couple of years, this thing has landed in our midst, which helps us orientate ourselves back to the Guy's campus. It's a good way of finding it. So here. I'd also just mention, for the sake of completeness, we've also got King's College Hospital down at Denmark Hill, I don't think that it's unlikely you'll be taking modules down there, but some of you who may take optional modules in perhaps psychiatry might end up down at that campus because we've got an Institute of Psychiatry down there that offer some modules. So you can unrivaled location. So again, just to give you a bit more detail of our Waterloo campus, I said we teach some of the biosciences here. There's also a lot of nursing and midwifery and social science public policy, so geographers, management, those kind of things. The Guy's campus is really the bulk of the biosciences plus medicine and dentistry. There's some medicine at St Thomas's, and the Strand, law, humanities, natural and mathematical sciences, and a bit of SSPPP. And one of the other reasons I put this slide up here is really to emphasize to you that King's is very much a multidisciplinary institution. It's a multi-faculty institution. We've got people here studying a whole range of subject matter, from the biosciences through to law, ancient Greek, etc. And we deliberately try to encourage students to mix in this kind of multi-faculty environment. So for example, those of you who will be staying in halls of residence, we don't segregate our halls of residence to put all the biomedical scientists or the medical students on the hall of residence near this campus. We actually mix you up because an important part of being at university is starting to get different perspectives on world problems, so bioscience problems. Talking about genetically modified crops, you may have a scientific, rigorous understanding of what goes on in genetically modified crops, but it's always good to be speaking to someone studying French and get their ideas. Say, well, why are you afraid of genetically modified crops? So because you've got to get that kind of world perspective. It's also never too early to get to know a lawyer, so these lawyers will also. Just to give you an idea of some numbers, King's College London in total has got about 25,000 students. It's students from all over the world, 145 countries at the last count. And that's roughly 15,000 undergraduates, 10,000 postgraduates. Within School of Biomedical Science, or Bioscience Education as we call it, we've got about just over under 3,000 students in total, 2,200 undergraduates, 500 postgraduates. So you will primarily be on the Guy's campus, I've already mentioned, and these are all the degree programs that we teach on the Guy's campus. And I think today we've got some biochemists, some neuroscientists, some molecular geneticists, and biomedical scientists with us. There's some biochemistry, molecular genetics at the Waterloo campus, but I'd say it's a relatively small amount, just a few modules. And what we don't expect is students to walk between the two campuses on any given day. So if you're doing a module that's run at the Waterloo campus, the way we organize our timetable is you'll be at the Waterloo campus for the whole day. So it may be Tuesday throughout the semester, for example. So you're not expected to go between campuses during your working day. We also teach pharmacy at the Waterloo campus. I don't think there's anyone here from interested in studying pharmacy, but that's another degree program. It's a professional program that we offer over at Waterloo. So as I mentioned, our graduates are highly thought after, and we get various accolades for this. And one of the other great things about our graduates is the gowns. So these, about 5-6 years ago, King's College London got permission from the University of London to award its own degrees. So before that, when you got a degree from here, you got a University of London degree, and University of London BSc in biochemistry or whatever. But we gained permission to award our own degrees, and as part of that, we had to design our own graduation gowns. And these graduation gowns, a friend of a friend of a friend of a friend of the principal knew Vivian Westwood, and she designed our graduation gowns. So for one day when you graduate, you will be wearing Westwood. So something to do. Now, as I mentioned, our graduates are highly sought after, and as I say, one of my key aims is to get more scientists into traditionally non-science-based careers. And we're having some success in this, and this is just a far from complete list of the kind of careers that our recent graduates have gone into. And some of them are kind of typical of what you might expect of a scientist. But you can see also we've got someone here's a sales trader in Morgan Stanley, a strategy analyst, we've got some management consultants, we've got some accountants, we've got a whole host of different careers that science graduates are going into and doing very well in them. And we get some of these people to come back and speak to our graduates at various careers evenings. And as I said earlier, one of the critical things we do is we need to emphasize that the kind of skills you learn as a scientist will set you up whatever career you decide to follow in. So by all means, those of you who are keen to follow a career at the lab bench, it's a great degree to do that. But equally, if you're concerned that your degree program you're not quite convinced that you want to spend your life working at a lab bench, doing a degree in the biomedical sciences will help you follow a whole host of different career paths once you graduate. Okay, so that's something in general about how we run our programs and why we think our programs are good. What about when you arrive here? Now, I'm just going to say a few words about what we call the Common Year 1. Now, as the name suggests, Common Year 1, what it means is all our students who are studying bioscience or biomedical programs, biochemistry, pharmacology, etc., they will study together in a common curriculum in year 1 of the degree. At the end of year 1, they can then head off in different directions depending on what degree program they're studying. So we have this common year 1 in first year. So why do we have this? Why do we take this particular model? Well, the first thing is that we know from a kind of we know that really to be whatever bioscience you decide to study, to be an effective bioscientist, you need to have an appreciation of the whole range of disciplines. So I'm a pharmacologist, I'm interested in drugs and drug action, but I can't be an effective pharmacologist unless I know something about biochemistry, something about neuroscience, something about physiology, something about pathology, because you really need to kind of see how all those biosciences fit together and understanding the body, the human body, to understand how drugs work on the human body. So we know that really to be an effective bioscientist, you need to have an appreciation of all the different arms of disciplines. And so what we do is we deliver kind of core knowledge and skills that are needed by all students of the biosciences. You may say, well, I don't need to know a lot about pharmacology because I'm here to study anatomy, and that's absolutely true. You don't need to know a lot, but you need to know something about pharmacology so that when you come across some of the kind of jargon associated with pharmacology in later years, it doesn't scare you, doesn't hit you, and you've heard something about it. Another great advantage of the Common Year 1 is it allows students to try new subjects before they commit to a specialised programme. We know, for example, that, again, I use pharmacologists just because it's my own discipline. We know, for example, there's very little pharmacology in the typical A-level curriculum. So students don't really know, well, I'm not really sure what pharmacology is. I'm not sure whether I'd like being a pharmacologist. So it's very difficult to ask them to commit at the age of 17 to becoming a pharmacologist. So what we say is at the end of this year 1, you've all done the same modules in year 1. You've all tasted all these different disciplines. And even if you've arrived here to study anatomy on day 1, if during that first year you decide, actually, now I've tried pharmacology, I really think I'd quite like that, you can change your degree program. So there's a huge amount of flexibility within these common year 1 programs for students to change between programs at the end of year 1. So don't feel that you've committed, having chosen to study biochemistry, you're not committed to leave here as a biochemist. That flexibility is built into the curriculum. Another advantage is it's simple. Everyone enters year 2 with the same knowledge and skills. We have students from lots of different countries and they're coming from lots of different educational backgrounds, and despite the fact that on paper these kind of background qualifications may be equivalent, we know that there are different emphases on them. So one of the things that we can do by the end of year 1 is we can get everyone up to the same speed for year 2 and year 3 modules. We can tie all these things together in a kind of nice coherent package so that when you're hearing about, for example, cardiovascular disease, you'll be looking at the physiology of the cardiovascular system, the biochemistry of the cardiovascular system, the pharmacology of the cardiovascular system, all in one package. So we can kind of mesh those different aspects of the discipline together into a nice package for you. And finally, as I mentioned, it's flexible. Now, these are the degree programs that are within the Common Year 1. So these ones here are all within the Common Year 1. And what that means is, if you come here to study neuroscience, you can switch to medical physiology. Biomedical science, you can switch to neuroscience. So if you're here to study any of those, you've got that flexibility. The only health warning I'd give you is if anyone's here by mistake wants to become a pharmacist, you must follow the MPharm degree. There's no transfer from these programs into the MPharm degree. That's because the pharmacy degree is regulated by the General Pharmaceutical Council, and they tell us what our students must be taught from the day they enter to the day they leave. And so there's really no flexibility within that program. So if you want to be a pharmacist, I just say you must register for an MPharm degree. There's no transfer between these ones. Disadvantage of the Common Year 1 is, first of all, you may say, well, I don't care what you say. I'm not interested in pharmacology. Well, you will be. The second disadvantage is that some of the classes are large. So the lecture theatres are in the lectures, because we've got all these students together, the lectures are big, and there's no getting around that. The way we get around that problem is by taking the big lectures for the kind of getting across the basic information, but then breaking students down into smaller groups for other types of teaching sessions. So for example, laboratory class practicals and workshops will typically have 50 to 60 students in them. We have smaller tutorial sessions where you have one tutor with 15 students addressing particular aspects surrounding material taught in lectures, material taught in practicals. We also make sure that the tutorials you will be in the tutorials alongside other students following your particular degree program. Because obviously if, for example, I think we've got about 10 students who might come to study medical physiology. If those 10 students are in a class of 450, even by Brownian motion, they may never actually collide together if we just left it to random. So what we do is we make sure all the physiologists are in the same tutor groups so that they get to know each other. So you will be taught in these small group sessions alongside students studying the same degree program. And then finally, these aspects deal with the academic aspects, but we also have a pastoral support where you have what's called a personal tutor, and his or her role is really to help you gain advice or to support you in a non-academic way during your studies. And your personal tutor and program advisors, they will also be from your home department. And I have, for example, 6 personal tutees who I look after from the Common Year 1 program. So I see a small group of students as a personal tutor. So we break these large classes down into smaller groups, and so we get around this potential problem of having large lectures. These are the modules. The reason I put this slide up is just to remind you that all our programs are modular based. Each year, to gain your degree, you have to study 120 credits, and all our modules are made up of either 15 or 30 credits. In final year, there are a few 45 credit modules. So if you add all these numbers up, you get to 120 credits. So each year you will be studying 120 credits. To progress to year 2, you've got to pass 120 credits of year 1. To progress to year 3, you've got to pass all. So you've got to pass gain all your credits to progress from year to year. Now, in terms of the way we structure these modules, I'm not going to go through each individual module, but we kind of start at the molecular and work out to the whole system. So we start by modules that look at chemistry, biochemistry, genetics, and molecular biology. Whether we like it or not, we are simply bags of chemicals. Everything we do, from you sitting there looking at that screen, to thinking about it, to think about how long ago it was you had your lunch, from your gastrointestinal system telling you it's a long time since you had your lunch, it's all driven by chemical interactions. The making and breaking of bonds. Now, you don't need to be a chemist to study the biomedical sciences, but you need to have an appreciation of chemistry so that when you hear things like that's an amide bond, you don't go, ah. So you've just got to, I say you don't need to be an expert in chemistry, but you don't need to be afraid of chemistry. So we look at chemistry. We look at chemistry in the kind of the biological setting, so what we call biochemistry, so how signaling goes on inside cells. So within the cells of our body, there's lots of different metabolic pathways that are talking to each other, how that communication happens. We look at genetics, because clearly what happens is we start off with the genome, the DNA, and that gets translated into these very complex and large protein molecules, and it's these proteins that do the business. All our enzymes, for example, are protein molecules, quite complex proteins, and the way they are folded is critical. And one of the things that we know is that many diseases these days are caused by mutations in this genetic material. So we know, for example, that if you get a mutation in your DNA that leads to a protein being misfolded because an amino acid in that protein goes from having a positive charge to a negative charge by mistake, then if you think about sticking a negative charge into a protein structure as opposed to a positive, that can change the whole shape of the protein because it starts to repel neighboring amino acids when it used to attract neighboring amino acids. So very simple molecular changes at the level of DNA can lead to a complete change in misfolding of the protein. We know that the most common form of cystic fibrosis, for example, is caused by one single amino acid being missing in a protein of about 4,000 amino acids. So it can be very, very kind of very profound effects, very simple changes at the molecular level. So these are the kind of things we look at in chemistry, biochemistry, genetics, and molecular biology. So these kind of things are all happening inside the cell. We then kind of pull back in our microscope and we start looking at the cell, what we call cell biology. How do cells come together to form different tissues of the body? Now we do this with particular focus on neuroscience because we've got a kind of a research strength in the area of neuroscience at the college. So rather than trying to cover every single cell type in the body, we focus on neuroscience, but the kind of underlying principles are the same. So how does a cell in the embryo know to eventually become part of your brain as opposed to part of your kidney or your liver? So we've got 3 different tissues here. This is kidney, I think. This is skeletal muscle. These are neuroscience. You've heard a lot about stem cells. And on the news, if you listen to stem cells, they are going to revolutionize our understanding and treatment of disease. And that is absolutely true. But we're still, to some extent, in the foothills of how we do that. What we don't quite know is if you take a stem cell, this cell that's got the potential to become any cell type in your body, kidney, muscle, nerve, what is it that drives it in one direction as opposed to the other? What combination or soup of chemicals must that stem cell be exposed to make it decide to become a kidney cell as opposed to a nerve cell, as opposed to a muscle cell? We've got some idea, but we're still not there yet. And until we get that idea, it's going to be quite difficult to implant stem cells in the brain, for example, in patients with Parkinson's disease, and make them become the nerve cell that's degenerated in patients with Parkinson's disease. In theory, we could do it, but how do we don't quite know what we have to do to control those cells to drive them in that direction. And that's the kind of thing that cell biology and neuroscience will be looking at. So we're going to special emphasize in nerve cells. We'll look at cell signaling, cell movement, and cell division. How do we control the development of the brain and the nervous system from the embryo, for example. And you also do a bit of lab work, which is looking at, well, how do we examine tissues down a microscope? What do we have to do to prepare tissues for a microscope? And can you, when you look down a microscope, identify the different types of tissue you see? We then pull back further on our hypothetical microscope away from cells and look at, well, how do all these cells and tissues come together to form functional biological systems? So here we've got the cardiovascular system with our heart and our blood vessels. How do all these things come together to allow your circulation to function? Now that's what we might call physiology. So there's a bit of it's anatomical, the kind of looking at it and say, well, how does the heart, what structures of the heart allow it to function as a pump? How is the heart developed in a kind of structural way to allow it to function as an effective pump? So that's kind of anatomy, I guess, would be the way to look at it. The other aspect is, well, how does that pump and all its connections, the vasculature, how does that function as a circulatory system to get oxygen to your tissues and remove carbon dioxide from your tissues? How does that function? And so we look at all these various systems, the musculoskeletal system that moves your muscles, renal system, etc., and just go through the different cell types and tissues involved in regulating those systems. So that's what we might call physiology, when things happen normally. Related to that is pathology. What happens when disease strikes? What happens in our vasculature in a patient with hypertension, high blood pressure, for example? So we compare the normal physiology with the abnormal pathology to try to see what has caused the disease, to try to get clues as to what might have gone wrong in those tissues. Now, as a pharmacologist, my job is to look at the normal, look at the abnormal, physiology, pathology, and say, well, is there any way that I could use drugs to convert this pathological state back to the physiological state? Now, sometimes that is relatively easy. Sometimes, in relatively few cases, do we cure disease. We can treat the symptoms of disease. So asthma, for example, we can give patients drugs that will relieve the bronchoconstriction they feel during an asthmatic attack, but we're not curing asthma. We're relieving the symptoms of the disease, but the underlying disease is still present. Similar with high blood pressure. Many of an increasing percentage of our population suffer from cardiovascular disease. high blood pressure. We can lower the blood pressure of patients with drugs. And those patients tend to be on these drugs for life to keep their blood pressure low. But the drugs don't cure high blood pressure, because you take the drug away, the blood pressure goes back up again. So we're not curing the disease, we're treating the symptoms of the disease. And so there's a lot of work to be done to try to understand disease so that we can actually produce curative drugs rather than simply symptomatic relief. And it may seem strange that we still don't know what goes wrong in the muscle cells of a blood vessel in a patient with high blood pressure. We know there's something wrong. The blood vessels are narrower, they're constricted, and we know that the muscle cells that surround the blood vessels are constricting far further than normal, but we don't know why. We don't know what has changed within them. And that's what some of the research in our cardiovascular research centre here is looking at. When it gets to disease of the central nervous system, the brain, it becomes even more problematic. This is again tying in with neuroscience. If we look at neurodegenerative conditions, Parkinson's disease, Alzheimer's disease really of the 20th century. We're living longer, and some of these diseases are becoming more prevalent because we're getting to the age where they start to appear in the population. But we also have disease such as anxiety, depression, schizophrenia, where we don't know what changes in the biochemical milieu of the brain of a patient with anxiety, for example. We really don't understand enough about the brain to be there. I spent my life, no, that's long. I spent my PhD, it seemed like a lifetime, studying a particular nerve cell in the brain, recording from it. And I could bore you for hours on the electrical activity of this brain, how the electrical activity changes in response to drugs. I was interested in drugs like morphine that produce analgesia. But how we go from the activity of that single nerve cell and multiply it up a billionfold to understand emotions such as love, hate, even pain, we have no ideas. We're not even in the foothills. We're not in the molehills at the bottom of the foothills of understanding how emotions like love, hate, anger are actually manifest are actually produced within the brain. So there's a great interest there. As far as diseases are concerned in pharmacology, one of the problems we have is we don't have great models for understanding human diseases. Whether we like it or not, rats tend to regulate their blood pressure in much the same way that we do. So if I have a drug that lowers the blood pressure in a rat or a mouse, there's a pretty good chance it would also lower the blood pressure in a human. But I wouldn't recognize an anxious rat. I wouldn't recognize a depressed rat. So how could I possibly start developing drugs that are going to relieve depression in the rats? So there are lots of problems with diseases of the central nervous system. Finally, so that's the kind of science you will be covering, from the molecule up to the cell, to the tissue, and pharmacology. We also look at various skills, things like mathematics skills, the ability to stand on your hind legs and communicate your ideas. Any scientist that doesn't communicate may as well not have a job, because we've got to be telling people about why we're doing it and what we've found. One of the things that's changed since I did my degree is the amount of information out there. There is a huge amount of information at your fingertips at the press of a computer button, and how you actually filter that information and filter out the wheat from the chaff, the good from the bad, is actually a skill set in its own right. And so we help you sort that kind of thing out. And also how to study at university. It's a very different way of studying compared to at school. You've got to become independent learners, because when you leave here, you've got to be continuing that independent way of learning, and so you need to develop your study skills. So we have a module on skills for bioscientists which cover all these things. So at the end of year one, we have various talks on course advising and options open to students in second year. You can talk to your personal tutor or your program advisor. Choose your year two modules, and depending on which degree program you're following, you have more or less choice. You could, for example, change your degree program if you wished, so you've got that flexibility. You take your exams in May, and all being well, you have a great summer. And that's where our students are at the moment. They're somewhere in here. Their exams, they're on holiday at the moment. They left, term ended on Friday. So their exams start in May. They'll be back in then to take their exams, and hopefully they'll all be off doing wonderful things over the summer months. So that's it from me. Just a taste of the way we structure, of what you'll be doing, certainly in your first year, and the way that the school is structured. I'm happy to take any questions. I think we've got maybe 5 minutes for questions if anybody's got any. So over to you. Yes?
How many exams could you expect to take in one month?
So the question was, how many exams do you expect to take? So all our modules the majority of our modules are a mix of coursework and end-of-year examinations. The actual contribution of those two varies slightly. If you've got a module which is very laboratory-based, it might be mainly in-course work rather than exam work. In fact, some of our final year project modules are completely in-course based, so you go and write a dissertation rather than sit in examination. But in year one, for example, typically you'll have 70%, 60-70% of your marks in a module will come from the examination, 30 to 40% will come from in-course. So there's a kind of continual assessment going on through the year. And each module, whether it's a 15 or a 30 credit module, will have an end of module examination. So if you take in year two 8 15 credit modules to get your 120 credits, you'd have 8 examinations. If you took 4 30 credit modules, you'd have 4 examinations. And one of the things we do at course advising is advise students and get them to think about the exam load they're taking on when they're making that decision. So each module will have typically an examination at the end of it. Any other questions?
Do you have the same personal tutor and program advisor through all 3 years, or do you only have them in the first year?
No, okay, so the question was, do you have the same personal tutor and program advisor throughout 3 years? Personal tutor, yes, unless you want to change him or her. So we have to accept that personal tutoring is a personal thing, and sometimes you don't get on with your personal tutor. So you, a student, can request to change the personal tutor, but we won't request to change you. Course program advisor, it depends. If you change your program, you will change program advisor because, so yeah. So personal tutor, yes. Program advisor, it depends. Any other questions?
Do you also work closely with medical students as well?
Not really. Okay. So certainly in early years, no, because the medical program and the bioscience programs, although they're sharing much of the same material, they're taught quite separately. I won't go into the differences between them, but I teach medical students about pharmacology, I teach bioscience students about pharmacology. It's quite kind of different aims of the session. The exception to that is in your final year, because medical students have the option of doing what's called an intercalating BSc, where they step out of the medical program after 2 years and do the final year modules that you will be taking. And so we typically have somewhere about 160 intercalating students in the school each year. So they would be, you'd be mixed in with medical students then. But they're not studying medicine at that point. They're studying neuroscience or physiology or pharmacology to get this intercalating BSc. Having said that, on this campus, there will be 400 medical students a year. In the union, there will be 400 medical students a year. So outside the confines of the lecture rooms, in the library, in the computing rooms, in the union building, you will be mixing with medical students. Sports teams, all that. Yes?
How varied are the second and third year courses and modules? Do they cross over a lot with the other bioscience?
Yeah, so the question is how varied are the second and third year modules? So we have about, I think we've got about 40 year 2 modules to choose from and about 70 year 3 modules. The modules tend to get smaller and more specialised as they become more advanced. And they're all kind of arranged around our main disciplines, which are neuroscience, anatomy, pharmacology, physiology, biochemistry, genetics, immunology. So they're all kind of run in those kind of disciplines. So there is, and one of the way we organise it is that when students are choosing their particularly their year 3 modules, there will be prerequisite modules they must have taken in year 2 to prepare them for that year 3 module. The prerequisites for all year 2 modules is the common year 1, but when they're choosing the year 2 modules, they've got to keep an eye on where they want to be at the end of year 3, because they need to make sure, for example, if they want to do a module in psychology in year 3, they have to take the prerequisite module in year 2 that will lead into that one. So again, that's what the course advisor helps the students with, thinking about that kind of decision-making process. But they all do slot together in kind of themes, I guess, would be the way to look at it. Any other questions? No? Oh.
Do modules change each year? So
Yeah.
Will that be available when you're on the digital?
Yeah, we don't, so I guess in terms of the actual module, the module will likely be running. I mean, every now and again, if someone, for example, who's teaching a very specialised module in year 3 leaves, we might not have the expertise to deliver that module, so that module might no longer run. But in years 1 and 2, the modules are pretty constant. What might change, though, obviously, particularly again in year 3, is the content, because science moves forward. So the content of a module, a year 3 module this year, will hopefully have advanced somewhat by the time you come to study it later. So the actual content may change subtly, but actually the module itself is unlikely to change. We have maybe 1 or 2 modules a year that change, but it's not there are many years when we don't have any changing. Sorry. Sorry.
If you study abroad, I understand that the modules might change a little bit. So
Study abroad?
abroad, yeah.
Yeah.
So if you're in your second year and you want to do modules that will lead on to the third year,
Yeah.
would studying abroad sort of narrow that?
Yeah, so the question is about, so we give students the opportunity to spend their year 2 at one of our partner institutions in Karolinska, California, Melbourne, various other places. We encourage students to do that. I guess one of the risks is that we can't guarantee that the curriculum followed at the National University of Singapore is precisely the same as here. But again, we give you advice before you go out and try to get a module package together that matches up as much as possible. And you may have to do a bit of reading over the summer months before you come back just to catch up. But it's not really been a big problem for our students. I think you gain far more out of studying abroad than the kind of disadvantage of not quite meshing those things up.